Hello everyone, welcome to my channel Anupma Biology classes. In this biotechnology series, today our topic is transgenic organism. This video is taken from CBSE class 12th, part 4th of chapter 12th. You have learned the basic principles of biotechnology and the process involved in it in the previous videos. You have also learned that biotechnology deals with industrial production of a large number of biopharmaceuticals and production of genetically modified animals, microbes, and plants. Let us now learn what are transgenic organisms and how they are used to improve the quality of human life. Now, here you can see the lecture overview that what you will learn in this video. So in this video, we will see transgenic organism in which transgenic animals, transgenic plants, transgenic microorganism, then biotechnological applications in agriculture, in which improvement in photosynthesis, herbicide tolerance, insect pest resistance in transgenic plants, and examples of the transgenic plants. Now, in this video, our first topic is transgenic organism. So, what is transgenic organism? Transgenic organism which contains functional foreign gene experimentally introduced into their genome by genetic engineering from another species. This is also known as transgenics. The foreign genes alter the host cell genetically and is termed transgene. The production of transgenic organism is known as transgenesis. The transgenic organisms are also called genetically modified organism, that is GMO. The latter may also be formed by introduction into them an extra copy of their own gene or a cloned and modified gene. Transgenics may be animals, plants, or microorganisms. Now, the question is, what is GMO and what is GM food? So, genetically modified organism can be defined as organism, that is plants, animals, or microorganism in which the genetic material, that is DNA, has been altered in a way that does not occur naturally by mating or natural recombination. Now, what are the possible benefits of GMO? So, there are many possible benefits like more nutritious food, disease and drought resistant plants that require fewer environmental resources, less use of pesticide, increased supply of food with reduced cost, and longer shelf life, and faster growing plants and animals. So, these are the benefits of the GMO, that is genetically modified organism. Now, here, transgenic animals. The first transgenic animals were produced in 1982. These were mice having in them the growth hormone gene from rats such as transgenic mice grow to double the normal size. Since then, many transgenic animals have been produced. These includes the following. First, in this picture, you can see transgenic sheep. Transgenic sheep having different foreign genes have been produced. Female sheep having human anti-hemophilic factor 9 gene in their genome have been produced. They secrete human clotting factor 9 in their milk. The factor 9 can be separated from the milk and administered to the hemophilic patients. The sheep flock can now provide enough clotting factor to supply all the world's hemophiliacs. Sheep containing bacterial genes which code for the enzyme needed for the synthesis of amino acid cysteine have been produced. They give higher wool yield. The amino acid cysteine is an important component of the protein keratin of wool. Sheep with gene for growth hormone have also been formed. They yield more mutton. Now, transgenic goats. Goats having a human gene that codes for a blood clot dissolving protein, that is tissue plasminogen activator TPA, have been formed. They secrete this protein in the milk. This protein is used for the treatment of coronary thrombosis. Now, transgenic cows. Cows containing cDNA coding for the antibacterial human iron binding protein, that is lactoferrin, have been produced. After this, transgenic pigs. Pigs having bovine growth hormone gene or human growth hormone gene have been formed. Such pigs grow a little faster and have greatly reduced subcutaneous fat. They can thus provide leaner pork with lower fat content. Transgenic pigs having human genes carry human antigens in their organs. The organs like heart, kidney, pancreas of such pigs can be transplanted into humans without the risk of rejection. Now, transgenic fish. Transgenic fish of many species such as common carp, catfish, salmon, goldfish have been produced. They contain human growth hormone gene. They attain a size twice of that shown by non-transgenic fish. Now, transgenic mouse. One strain of transgenic mouse contains a human gene that causes breast cancer. This enables the researchers to study the very early development of cancer. Transgenic mouse carrying a gene for human growth hormone 2 grows to more than twice the size. A plasmid carrying human growth hormone gene is injected into the egg that develops into a super mouse. The human growth hormone gene produces human growth hormones in the cells of the mouse that therefore grows to an unusual size. Now, what is molecular farming? The method of manufacturing drugs in transgenic animals is called molecular farming or molecular farming. The transgenic animals can revolutionize the pharmaceutical industries by providing large quantities of the valuable drugs. Transgenic technology permits the rapid introduction of new traits. Here you can see the process of molecular farming, that is candidate enzyme, that is the structural gene, then plant expression vectors, then introduction into the plant, then transgenic plants, then plantation, then harvest and fresher formulation. So this is the way of molecular farming. Now, after this, transgenic plants. It is easier to produce transgenic plants than animals. A single cell in most plant species can regenerate a whole plant. Thus, a single genetically engineered cell can produce a new plant with new traits. Now, what are the potential applications of the plant genetic engineering? There are many applications like plant genetic engineer may produce transgenic plants that are resistant to disease resulting from viral, bacterial and fungal infections, pests such as nematodes and insects and pesticides. Can tolerate adverse environmental conditions such as drought, high temperature, frost, salinity, heavy metals, etc. Can produce pharmaceutically important compounds such as human insulin, interferons, hormones, blood clotting factors, etc. Can synthesize antibodies and vaccines, and their fruits when eaten may give immunity against disease in children. Can fix atmospheric nitrogen and exclude the need for using costly fertilizers. May bear flowers with new colors and long life for the better ornamental prospects. May yield slow ripening fruits for the longer shelf life. So these are all about the transgenic plants. Now, the last is transgenic microorganism. So, what are transgenic microorganisms? Many kinds of new genes have been introduced into microorganisms for various purposes, like transgenic bacteria and yeast. That is, bacterial and yeast cells have been engineered with the new genes that code for using human proteins. Such proteins include insulin, blood clotting factor, growth hormone, tissue plasminogen activator, interferons, interleukin, and tumor necrosis factor, Now, transgenic bacteria with the environmental use. So, genes have been introduced into bacteria to create bacterial strains having environmental important uses. So, such bacterial strains accelerate the degradation of oil spills, destroy dangerous chemicals, and transgenic E. coli. So, here you can see the transgenic E. coli, engineered E. coli produce bovine growth hormone. This hormone, when injected into cows, raises milk production as well as beef output. Here you can see that transgenic bacteria and yeast, and the process like the desired gene is identified from the donor cell, then the gene is inserted into a plasmid, that means in the bacterial cell, then formation of recombinant plasmid, and then the recombinant plasmid is put into a bacterial cell. If you talk about the insulin needed for the treatment of diabetes, and because of this human insulin production, you can see here that how the human insulin form by the recombinant DNA technology. If we talk about the growth hormone needed for the treatment of pituitary dwarfism and the blood clotting factors used in the treatment of hemophilia. Here you can see the gene targeting for the knocking mouse. So what is gene targeting? A variations of the transgenic technology is gene targeting, in which a multicellular organism has a particular gene inactivated in each of its cells. By observing these organisms, researchers can pinpoint the roles of particular genes. Here you can see that mouse genome, then linearized targeting vector, then knocking allele. No essential proteins production has been compromised, but gene X is now being transcribed at the locus. Then generate chimeras and then breed mice with the knockout allele. So this is the gene targeting. Now, after this, what are the biotechnological applications in agriculture? So, our conventional methods of the crop improvement, such as selection, hybridization, protoplast fusion, involve the whole genome of plants. However, the latest recombinant DNA technology or the genetic engineering involve transfer of one or more genes, that is DNA fragments, from one plant to another. The plant in which foreign genes have been introduced is called transgenic plants. There are two distinct ways here or techniques for introducing foreign genetic material into the plant cell genome. Before this, what are transgenic plants? So, the modification of DNA using genetic engineering techniques aim is to introduce new traits to the plant. The inserted sequence is known as the transgene. The purpose of inserting a combination of genes in a plant so as to make it as useful and productive as possible. Examples in food crops include resistance to certain pest disease or environmental conditions, reduction of spoilage or resistance to chemical treatments, or improving the nutrient profile of the crop. Examples in non-food crops include production of pharmaceutical agents, biofuels, and other industrially useful goods as well as for bioremediation. So, if we talk about the techniques for introducing foreign genetic material into plant cell genome, there are more ways in which two are common. The first, through a vector, requires selection and isolation of the desirable fragments of DNA, hooking it to a vector, that is the bacteria, Agrobacterium tumefaciens, a virus or any other vector, and introducing novel genes into the plant cell. So, the second, through direct introduction of DNA, involves co-cultivation, that is, culturing the recipient protoplast with purified DNA, or electroporation, that is, application of electric impulses to change the porosity of the protoplast so that they directly imbibe the purified DNA, and microinjection, that is, direct injection of DNA with the help of a micropipette. Now, in this applications in agriculture, now The improvement in photosynthesis by biotechnology. So what are the improvement in photosynthesis? Ribulose 1,5-biphosphate, that is Rubisco, is an important enzyme of photosynthesis which can act both on CO2 and O2. Under certain conditions in light, the enzyme Rubisco oxygenated ribulose biphosphate and inhibits the carboxylation, as a result of which 25% of the fixed carbon dioxide is photorespired as carbon dioxide. It is now possible to isolate the Rubisco gene, modify it accordingly, and reinsert into the plant to minimize the loss due to photorespiration. One such gene controlling Rubisco enzyme has been transferred from pea plant to tobacco plant to increase photosynthetic process and high leaf yield. So this is improvement in photosynthesis. Now, what is herbicide tolerance? So if you talk about the herbicide tolerance, herbicides are the chemicals which kill the weeds. When these chemicals are sprayed in the field, they not only kill the weeds but also damage the crop plants. In recent years, genetic engineering has helped to develop such transgenic crop plants which are resistant to herbicides so that they are not damaged when farmers spray herbicides in the field. Some herbicides resistant crops produced by genetic engineering are tomato, potato, tobacco, soybean, cotton, corn, oilseed, rape, sugar beet, petunia, etc. Herbicide resistant plants have been developed in such a way that they continue to produce normal crop yield and at the same time remain unaffected by the activity of herbicides. This has been achieved by overproduction of those enzymes which are inactivated by herbicides, inducing mutations resulting in the production of enzymes which are less sensitive to the herbicide, introduction of such enzymes which causes detoxification or herbicides. Several such enzymes have been identified in plants as well as in microbes and transferred into the crop plants to produce transgenic plant resistant to herbicides. Thus, herbicide-resistant transgenic plants reduce the use of weeding labor, farmers' cost, and increase yield. Now, transgenic tobacco plants. Tobacco plants containing a gene from a bacterium Bacillus thuringiensis have been produced. This bacteria gene specifies an insecticidal protein that destroys the stomach lining of insects and kills them. The tobacco plants with these genes produce their own insecticide. Now, transgenic tomato and potato plants. Tomato and potato plants having viral coat protein genes have been formed. These genes are resistant to viral diseases. They produce small amounts of the viral coat protein which interferes with the ability of an infecting virus to shed its coat and replicate. So, if you talk about the transgenic potato, they are more tolerant to water deficit in the root zone, preserve more water in green tissues, and maintain chloroplast functions and have higher accumulation of chlorophyll B and xanthophylls than with wild type. Enhanced drought tolerance in transgenic potato confers greater tolerance to high light stresses, stomatal closure, and diminished carbon dioxide supply. Now, flavor saver tomato. Tomato plants have been engineered with an antisense gene that retards ripening. These genetically modified tomatoes plants are called flavor saver, produce much less cell wall degrading enzyme, that is polygalacturinosis, responsible for fruit ripening. Therefore, the fruits of these plants have much longer and more flavorful shelf life. Now, after this, insect pest resistance in transgenic plants. So, the day is not far off when our crop plants will resist all kinds of pests. Several such transgenic plants have been developed and many others are being experimented in the laboratories. One such plant is Bt cotton, in which the Bt toxin has been used as a biological insecticide. The bacterium Bacillus thuringiensis, that is Bt, is a common soil bacteria which produces a protein toxin that kills certain insects. The toxin is a crystal, that is cryproteins. There are several kinds of cryproteins which are toxic to different groups of insects. The gene encoding cryproteins is called crygenes. That is, the cry is protein and written in Roman letters in which the first letter is always capital, whereas cry is a gene which is written in italics and all the letters are in small. Biotechnologists have been able to isolate the gene responsible for the production of toxin and introduce it into a number of crop plants using agrobacterium TI plasmid mediated transformation. The new genetically modified Bt cotton is resistant to the bollworm complex, that is specific caterpillar pest. Tomatoes with Bt genes are completely protected from an attack of caterpillars. Similarly, the genetically modified tobaccos with Bt gene is protected from hornworms. Scientists are busy in developing transgenic rice plant resistance to stem borer, leaf borer, and caseworms. Biotechnologists have been able to obtain a number of Bt toxin genes. These genes have been cloned from bacteria and incorporated in many plants such as Bt cotton, rice, potato, soybean, Bt corn. The genetically modified plants having cry or Bt toxins genes do not require treatment of insecticides against a specific insect pest. Some common insects affected by the produced cryproteins are armyworms, tobacco budworms, beetles, and flies, mosquitoes, etc. The harmful insecticides proteins are produced by the introduced bacterial genes in inactive toxin crystalline form. As the insect feed on the genetically modified plants, the inactive protoxin crystals pass into insects' guts and, in the presence of alkaline pH and digestive enzymes, get converted into toxin. The toxin subsequently binds to the epithelial cells of the midgut of insects and results in swelling and lysis of midgut epithelial cells. The insect subsequently dies. Different crygenes have been found to be effective against different insect species. Now, here you can see the Bt cotton. Cotton crop is generally attacked by the caterpillars of the cotton bollworms, that is Aereas vitella and Aereas insulana. The caterpillars bore into top tender parts of the shoot, floral buds and balls. The affected balls bear holes. These open prematurely and have poor quality of lint. Insecticide treatment on bollworm infected cotton crop is very expensive. Genetists have isolated cry first AC gene and cry second AB gene and incorporated it in the DNA of cotton to develop genetically modified crop commonly known as Bt cotton. The Bt cotton contains Bt toxin gene against cotton bollworm. Now Bt corn. In the same way, cry first AB gene has been identified and introduced in the corn plants to form Bt corn to protect the crop from corn borer. Nematodes is a group of organisms which parasitize a large number of plants and animals, including and causes here human beings. One of the common nematodes is Meloidigene incognita that infects the root of tobacco plants and causes a great loss by causing a reduction in yield. These infestations were prevented by using a novel strategy which are based on the process of RNA interference, that is RNAi. RNA interference is a powerful reverse genetic tool to study gene function. RNA interference is a system within having living cells that helps to control the activity of specific genes. This method involves participation of a long double-stranded RNA to silence the expression of the target genes. Upon introduction, the double-stranded RNA enters a cellular pathway, gets processed into small interfering RNA, then single-stranded RNA assemble into RNA-induced silencing complex. Here, the siRNA subsequently, it means a small interfering RNA, guide the complex to complementary RNA molecules where they cleave and destroy the target mRNA. In this way, the entire process causes silencing of mRNA, that is, prevent translation of the mRNA. The source of this double-stranded RNA could by form an infection by virus having RNA genome or transposons that replicate via an RNA intermediate. The process of making tobacco plants resistance to nematode involving the following step, that is, initially the nematode's specific genes are introduced into the host plant using agrobacterium as vectors. Then introduction of DNA was made to produce both sense and antisense RNA in the host cells. Then the sense and antisense RNA were complementary to each other, so they form a double-stranded RNA. Then this double-stranded RNA initiated RNA interference and finally silenced the specific mRNA of the nematode. The transgenic tobacco plants have now become nematode resistant and prevent the parasitic attack of nematode. Such transgenic plants resistant to insects and pests offer many advantages to farmers. The primary advantage is production of labor-free insect protection of the crop plants at every stage of development. Secondly, restrict the use of pesticide, leaving the soil and groundwater pollution free. Now, after this, some examples of the transgenic plants. So there are some examples of the transgenic plants which are listed here. You can see the figure. Now, if we talk about the golden rice, so this is the transgenic plants and the useful characters are developed at Swiss Federal Institute of Technology rich in vitamin A. The rice grain are golden yellow in color. It contains beta-carotene gene for daffodil plant and also genes from some bacteria. Golden rice will prevent child blindness caused due to deficiency of vitamin A. Now wheat. Resistant against the herbicide PPT, that is commercial name Basta, 20% PPT. After this, Brassica napus. Transgenic plants are male sterile. The transferred gene prevented normal pollen development, leading to male sterility. In addition, a gene encoding hirudine, a protein that prevents blood clotting, is synthesized chemically and then transferred into Brassica napus. This gene is activated in the plant and it starts synthesizing hirudine, which accumulates in seeds. The hirudine is then extracted and purified to be used in medicine. Now, tomato. Resistant tomatoes which exhibit delayed ripening have been produced by using gene for SEC deaminase. These transgenic tomatoes are called flavor saver tomatoes, which carry an artificial gene that gets transcribed into an antisense RNA. This results in the inhibition of ethylene production so that ripening of tomato is delayed, preventing rotting and spoilage. After this, potato. Starch content has been increased by 20 to 40% by using a bacterial ADP glucose pyrophosphorylase gene. Now, here you can see the picture of the GMO genetically modified potatoes. Now, here tobacco. So, CPTI, that is cow P trypsin inhibitor gene, has been introduced in tobacco to show resistant against pest. Now, genetically modified bananas. Transgenic banana contain in their genetic material a portion of the toxin gene of the E. coli. These bananas act as edible vaccines to protect children against diarrhea. And the last is Bt cotton. Transgenic cotton having Bt2 toxin genes isolated from Bacillus thuringiensis is resistant against bollworms. The government has finally agreed to allow cultivation of genetically modified Bt cotton. So this is the end of video in which we understood about the transgenic plants, animals and microorganisms and the applications of biotechnology in agriculture. Now, the upcoming next video in the biotechnology series will be on the biotechnology and medicine. So if you understand this video, please like and share it and subscribe to my channel Anupma Biology Classes. If you have any questions, any queries or any suggestions, you can ask in the comment section below. Thank you for the watching.